Good afternoon, and thanks everyone for joining our panel. I'm Geulah Livshits, a biotech analyst at Chardan. And so the low-hanging fruit for gene therapy has initially been rare disease and gene replacement, but as programs in those indications advance, the groundwork is really being laid for expansion into larger indications. And while this trajectory could translate to multi-billion-dollar market opportunities, in many cases it necessitates navigation of manufacturing, scale-up, and other logistics. So in this panel, we'll discuss issues relating to manufacturing for larger indications. It's my pleasure to introduce our participants today, starting with Dr. Josh Grieger, a CTO of AskBio, a subsidiary of Bayer, Dr. Shankar Ramaswamy, co-founder and CEO of Kriya Therapeutics, and Faraz, CEO of Tenaya Therapeutics. Thank you all for joining us today. The format for the session is roughly 40 to 45 minutes panel discussion. And if you have questions during the session, feel free to type them into the question box and we'll try to ask them on your behalf. So again, thank you all to our participants for joining us today. And let's start off with having each of you introduce yourself and your background in gene therapy. And we'll start with Josh, then Shankar, then Faraz.
My name is Josh Grieger. I've been in the gene therapy space for a little over 20 years now. I got my doctorate degree in Jude Smolsky's lab at UNC and have been working with him ever since through all the transitions, UNC, Bamboo, Pfizer, and now AskBio with Bayer, and got to see all the transitions from small scale manufacturing, academic manufacturing, now through all commercial scale manufacturing, and it's been a very interesting career. I'm very fortunate to be in the gene therapy space for as long as I have, working with Jude.
Great.
Hi, I'm Shankar Ramaswamy, co-founder and CEO of Kriya. My personal background, I'm a physician, was part of the early foundational team at Roivant Sciences and started Kriya in 2019, which I co-founded with Fraser Wright and Roger Jeffs, and excited to be here.
Great, and Faraz.
Faraz, Tenaya Therapeutics. I've been in the gene therapy space. I've been in the biotech space for more than 20 years. Of that, a little bit more than a decade now in gene therapy. Initially at Bluebird Bio, when it was a small preclinical company with 20 people. That was ex vivo lentiviral-based therapies for rare genetic disorders oncology. After a few years there, I went to REGENXBIO. So that was AAV gene therapy for CNS, ocular, liver-based applications, including rare and prevalent indications. And now at Tenaya Therapeutics, company focused on the heart, and where we're doing gene therapy, AAV-based regenerative medicine, and early efforts in gene editing as well. Pleasure to be here.
Great. So the focus for this panel, as I mentioned, is on manufacturing considerations for larger indications in genetic medicine. So I'd like to understand what each of you sees as the greatest bottleneck in terms of scaling manufacturing for such indications, and also whether you have any proposed solutions that could be employed to address some of these challenges. And let's start with Shankar and then go to Faraz and then Josh.
Sure. So let me just first state at the outset, the mission of Kriya is really to build a fully integrated engine in gene therapy that consolidates many of the critical elements that we see that are needed for design, development, manufacturing, characterization of gene therapies. And I think we're at a point in the field where we understand gene therapy well enough to really know some of the critical success factors and failure factors. And I think part of the challenge the field has had in its evolution is really a number of disparate data sets that have been generated and very niche focused efforts on specific parts of various problems of gene therapy. So the fundamental thesis that we have is to consolidate all of the critical infrastructure and technology that's needed to systematically bring new advances in engineering and scaling up of gene therapies and apply that across a range of indications, both rare and prevalent. So that's just at the outset, the core mission of Kriya. And I think as part of that kind of reflects some of the strategy here, which is, how we think about overcoming some of the key technical challenges in this field. I think there are challenges at the intersection of molecular biology, process sciences, and biological performance that really do require large data sets, iterative improvements in process that can be applied across multiple different products and shared learnings across those different products that allow us to move faster with each subsequent development effort. And I think we feel that if you have a broad enough pipeline and a platform process to really leverage that pipeline against, you're in a position to make more rapid improvements in each individual part of the process. Part of the problem that we also see is that the field has largely been reliant on many academic processes to date, historically, and some of those processes are antiquated and really not set up to scale. And so having continuity and a clear path to large scale, especially if you're going after bigger indications with larger addressable populations, that continuous path is critical to have success rather than having a disjointed development effort with various iterations of process along the way. So hopefully that gives you some sense of where our head is at, that having tightly integrated capabilities is critical, and having that continuity and platform in mind, from the start, even from early development, is essential to ultimately achieving scale.
Great. Faraz?
Yeah, I mean, actually, no disagreement here. A lot of what Shankar said really resonates with us. So Tenaya was founded in 2016 with an idea of pursuing next generation medicines for heart disease. So we are very focused on the heart disease, but within that, there's a range of things that we do. We have 3 distinct platforms, each of which are trying to solve a different problem in the field of heart disease. So we have gene therapy, we have regenerative medicine, which also happens to use viral vectors for delivery, as well as we have a modality agnostic drug discovery platform that we call precision medicine. So formally in our portfolio, we have AAV-based therapies and small molecule, and in the future, we may have other kinds of biologics or gene editing and even non-viral delivery. So we're quite broad in terms of modality, and then we have to be very thoughtful and careful as we think about manufacturing. Like Kriya, we are focused on both rare and prevalent forms of heart disease. And in our early pipeline, we have gene therapies for some of the leading genetic causes of genetic cardiomyopathy that can affect 70,000 patients, 100,000 patients in the U.S. alone. So those, even though they're rare and orphan, they're large, rare and orphan. They are much larger than some of the inborn errors of metabolism that some of our peer companies are going after. But then we also have programs that are intended to go after indications like heart failure with a reduced ejection fraction, or post-MI heart failure that affects millions of patients in the U.S. and certainly in the G7. So from the get-go, from the company's founding, we knew that we had to prepare for a world of large indications. That doesn't mean we've cracked all the nets, but we know that we had to start working on it if we wanted to have a hope of eventually having products that could be adopted. And so scaling up is really important, and it's really important to us, and getting to, and like Kriya, we have chosen to integrate all the different pieces. So we integrated non-GMP capabilities, GMP capabilities, novel capsid engineering, promoters and regulatory elements, drug delivery, all of those pieces under one roof, because we felt that it wasn't one solution to address the whole range of things that we're going after, but we needed each of these pieces as part of an integrated whole to then be able to come up with platforms that can address both rare and prevalent forms of heart disease. You asked what the greatest bottleneck is. I'm sure we're going to dive into it, but the ability to scale, ultimately we are limited by the manufacturing platforms that we have. And we have to achieve technical breakthroughs, not only in the product side, but on the manufacturing side, to be able to scale up to a degree that you can consistently produce enough drug and at commercially and economically viable prices with low batch-to-batch variability and low cost of goods in order to support a commercially viable product. So I'm sure we'll dive deeper into that as we go deeper into the conversation.
Great. Josh?
Yeah, I agree with both Faraz and Shankar on their feedback. For AskBio, we have a pretty large portfolio of programs. We have small rare disease, monogenic diseases, as well as larger indications such as Parkinson's and congestive heart failure. And we are, of course, a fully integrated gene therapy company, so we approach manufacturing scalability and manufacturing from multiple facets. One of them is through our platform manufacturing technology using the ProTen system. And so we've got over a decade's worth of experience with that. And we can manufacture different serotypes through minor modifications to the process to optimize the full output for each of the viruses. We also have a new raw material that we're investigating and putting in most of our programs. You may have heard of doggy bone DNA. So we're moving away from plasmid and going to a synthetic manufacturing of a doggy bone DNA. And this allows us to look at scalability. We can generate up to 2 grams a week of doggy bone DNA. And when the facility tab is up and functional, as it continues to grow, we have more pods. We anticipate being able to manufacture up to 50 to 75 2,000-liter bioreactors per year in our commercial manufacturing facility. And we're also looking at not just scaling up, but enhancing or intensifying our manufacturing process to yield more vector out of the bioreactor, and obviously, have a net to the clinic much higher. So from a doggy bone DNA perspective, we have a raw material that we can scale. We have 2,000-liter bioreactors in place in San Sebastian at Virogen Commercial. And we also have Virogen in Spain as well for Virogen Clinical. And so we have scalability from 50 to 500 in our clinical facility and up to 2,000 liters in our commercial facility that'll be coming online soon. In addition to that, we have capsids. So we work with the different approaches, capsid libraries, rational design. We acquired a company in Edinburgh, Scotland called Simpromics, otherwise known now as AskBio Europe. And so we're implementing synthetic promoters, tissue-specific promoters. And so the overall goal is to have scalability, increase the amount of output for each bioreactor, which would drive down cost, but then working with the optimal capsid, the optimal promoter, the optimal therapeutic cassette, which in the end would hopefully lower doses and then lower the impact on the demand for manufacturing and overall lowering costs. That's our goal. I think that's everybody's goal. And I think everybody's working towards that in their own way.
Great. So I think all of you touched on, scaling up in some way. So when is enough? There's as you mentioned, there's scaling out in terms of having multiple bioreactors. There's ways to make the process more efficient for the given bioreactors that are available, and then there's moving to larger volumes. So from your perspective, what bioreactor scale is needed to support large indications? Is 2,000 or 3,000 sufficient? Is there a need to go to 10,000 liters, or is it better to kind of scale out at current capacities? I'll leave that open, whoever wants can take that one first.
Sure, I'm happy to take an initial stab at that. I think that, it's a function of a little bit of unknown at this point, because no one has really gotten to the size of bioreactors and scale that you see with monoclonal antibodies and other biologics. So there's a bit of an unknown here. And I think part of the unknown is also what are the sizes of commercial opportunities that we're going after. The 2 commercial AAV products are, not prevalent where those types of, supply needs are existent. So there's a bit of an unknown here. I think that to have a chance to actually get to large scale bioreactors does require advances in novel technologies. So there are, and curious for Josh and Faraz's perspective on this, but there's probably, bioreactor limitations in size of what you can get to with conventional methods of production. And so you're necessarily going to run into some upper bound limits that are never going to allow you to get to 10,000 or 20,000 liter bioreactors with conventional processes. So you really do need to think about a continuous path from a next generation scalable process. And it starts with delivering that process at smaller scales and getting to the 2,000 or 3,000 liter scale. As Josh mentioned, that AskBio has, we're at our facility scaling up to 3,000 liter as the upper end of our current bioreactor capacity. So I think probably once you get into that zone, you've already addressed many of the populations that are currently on deck. If we're talking about a universe in the future where gene therapies are being delivered to millions of patients, you might not start to think about processes that allow for 10,000 liter plus scale production. But I think we're a while away from that type of need being there. Curious for others' perspectives.
I'll join. I agree with Shankar. I think 2,000 to 3,000 liters single-use bioreactors is probably where it's going to be for a little while, and then changes to the conventional methods, right? The virus infection add back and all those, generating enough some virus to scale. And then with transfection, obviously there's going to be limitations potentially on that. And so you may be forced to start looking at generating a producer cell line that can be activated through an addition of a small molecule type procedure if you go to these even larger scales. Things that we're starting to look at as well for some of our larger indications. So some indications may be good with triple transfection, double transfection, whatever methods you use, and other indications may require a switch to more of a stable producer cell line and potentially going to higher cell densities or even higher, larger bioreactors. So I think there's definitely going to have to be a technology shift at some point in the future when needed.
Yeah, I mean, I don't disagree with what either Shankar or Josh said. It's exciting to think about a world of 10,000 liter bioreactors, but I think we're far from that, and not only in terms of technical challenges, but also need, right? Even when we talk about large indications, I think we can't lose sight of what's the goal here. The goal, yes, eventually to hopefully treat millions of patients, but really what we're trying to strive for are precision medicine approaches, right? We're really trying to go after, find those genetically defined or otherwise defined subpopulations, because you're trying to actually get to, in our case, I guess, the rapid approvals for well-defined severe populations. And then you may need a different product to address a different population within that. So whether you're looking, even when you take a large population, you're really going to segment those into smaller pieces. At least that's what we see in heart disease. And then when you start doing that, then you're not trying to serve millions of patients with one product, but you're actually trying to serve them with multiple products. So we don't need to get to 10,000 liter today. We also need to think about the product profile and what's the likely adoption curve, and what does that curve look like for a one and done therapy versus for chronically administered therapies. So I don't see the gene therapy field anytime soon getting to the world of, our friends at Genentech, I can see from outside my window, who up in Vacaville have these, farms of bioreactors that are continuously running, and we just don't see that, because we're not going to have chronically administered therapies. These are one and done therapies. So the adoption curve is going to be different. And so part of it is actually not just solving technical challenges to scale up, which we need to do, but we also have to figure out what is the optimal configuration of bioreactors and supply chains to be nimble to respond to the ups and downs of various products that are going to come through your pipeline. You may go several years with a couple of rare diseases that you're launching, and what you need there won't be the same as what you might need if you're launching for something more prevalent. So I think some more thought needs to go into how do you flex, not overbuild, both in terms of scaling out and scaling up, but actually coming up with a model that works for the more realistic situation that we're going to have products emerging in a spotty fashion, a rare one here, a prevalent one there, and whatnot. So that's something that we think about. And I do think I think the field is still honestly in its infancy of understanding, we still can't quite understand why some patients are going on clinical holds, right? And the intersection of the product attributes, the capsid and the manufacturing. We're getting better. We have assays to get better and better. But I'd argue that we're still in our infancy of really understanding the product attributes and its link to product characteristics in vivo. And I think once we need to solve that and figure that out, so that when we're making changes, we understand the implications of a change here for this rare disease and how that's going to carry through for a more prevalent indication. So there's a lot of work to be done at the smaller scale first, partly to crack those nuts and understanding of the ideal product attributes, and especially in a world of novel capsids, novel promoters, all the things that Josh, you guys are working on, Shankar, it sounds like you guys are working on as well. We are as well. How does that change your product attributes? Therefore, how might that create some scale up challenges, or how might that affect your product attributes as you start scaling up? We don't have the answer to all those questions. So there's more work to be done. And I think that is probably the more important work before, without rushing up to, say, 10,000 liter or even 5,000 liter for that matter. We eventually have to prepare for that future, but that future isn't here now, right? So I think there's work to be done, and those who are thinking ahead and being patient and putting one step in front of another now, they will yield insight that will pay dividends 5 and 10 years from now.
Great. So earlier, I think all of you mentioned something around cost with regard to some of the bottlenecks. So I'd like to drill down on that a little bit more. What are the largest cost drivers in AAV manufacturing, and are there strategies that you're looking at that could mitigate costs, or technologies that you can implement to further reduce them? I know you touched on them a little bit, but again, I'd like to elaborate on that a little bit more. We can start with Faraz.
I think that's the conundrum, right? I mean, eventually you do need to scale up to get down to lower cost of goods, or you need to get higher productivity per bioreactor run, right? I mean, and you got to reduce overall dose. So the path to reduction of overall dose may be, and I think that's shared by all of the panels here, you may need to look at things like a novel capsid or a novel promoter, or things to boost the expression per virus delivered into a cell, right? Because that's actually the starting point of dose reduction, is reducing the amount of viruses that you're putting in the first place. Then of course, once you understand that and you've solved that equation, then you want to produce as much virus as possible per bioreactor run. And then, if necessary, scale up. So again, I would say the same things. We actually first need to identify the ways to reduce the overall dose per patient. And some of those are not in manufacturing. Some of that will be in research, including capsids and promoters, and even delivery closer to the heart, in our case, for example, delivery closer to the brain for somebody else. Once we reduce the overall dose requirements, then we have a better shot at reducing the overall cost of the therapy, and of course, burden on the patients and burden on the manufacturing system. Once you get to the manufacturing side, I mean, there's a variety of things I'm sure my colleagues will comment on, but I don't want to lose sight of that the fundamental driver to reducing costs is reducing dose, and the fundamental driver to reducing dose isn't manufacturing. It actually starts with your research and your product, including the capsid and the promoters and whatnot. So I'll turn it over to Josh and Shankar to add.
Yeah, no, I totally agree. Touch base on earlier about optimizing in the research side before it gets kicked over to CMC to make it is very important and challenging, right? So there's a lot of new technologies. There's a lot of competition out there. So there's a little bit of pushback on how much of this technology can I get in. I need to get in and see how my capsid performs in a human. And so there's a lot of going into the clinic, learning how your capsid performs, and then going back and figuring out how to make it better. Then you have the second generation, the third generation. Obviously, a lot of the costs that drive the overall product are things like media costs, right? Synthetic media is not cheap. Your starting materials, whether it's plasmid or you have the baculovirus system or that, and you have to characterize those at GMP level. All that's pretty expensive. Transfection reagents are expensive. So all of that adds up. And like Faraz says, as you scale up, that's when you start seeing the cost savings. And so it's, developing the best drug product you have at the time to lower the doses, and then understanding, the limitations of your production system. But I think one thing that it's obvious to everybody in this space is manufacturing history, right? That you can go from tox to clinic and only manufacture a handful of batches, and only one potentially sometimes at scale. And then the FDA and other regulatory authorities are setting the bar higher now. And you don't have enough history and enough analytics to set specifications for phase 2, 3. And so everybody's taking a step back, generating the reference standards, making sure their analytical methods are in place, well characterized, and having to generate additional manufacturing runs. I don't think it's uncommon that a number of people can get into phase 3 or pivotal with a handful of manufacturing runs. So there isn't a lot of data out there to really characterize your drug product, which can be a major setback when you go to BLA and commercialization. And so it's building that into your CMC timeline and there's added cost to that.
Yeah, not much to add from what Josh and Faraz said. I would say, just to reinforce what Josh actually just described, I think one of the biggest costs of development and manufacturing related costs are lost batches and unreliable scale up. And so every time you scale from a research scale to a 50 liter to a 500 liter to a 2000 plus liter, there are, uncertainties of each of those leaps that result in lost batches. I think, to step back and this may not be relevant necessarily to the panelists on the call, but I think that, when it relates to having external manufacturers of product, that can often result in certain inefficiencies as it relates to lost material from various tech transfers that need to happen and need to be executed successfully. So these are factors that do contribute to the overall all-in cost of getting the manufacturing right for a given product. And I think to Faraz's point, it's a function of the dose requirements. So I completely agree with what he said on needing to get the technical features of the product right to minimize the dose. I also think it's a function of picking the right targets where the dose is manageable. And so, we're talking about, sometimes these the dose numbers are reported in VGs. These are sometimes tenfold, hundredfold, thousandfold differences in doses. So you think about the different challenges required at manufacturing something 1,000 times more of that product compared to 1,000 times less. That's a big difference when it comes to cost and feasibility of delivering enough material. So I think those are some of the factors, but underneath all of that is just the operational complexity of doing this successfully at different scales and reliably scaling up.
And some of the other things, I just wanted to pick up on something that Josh said, right? It's like the starting materials are expensive, right? So, expensive in, expensive out. And why is that the case, right? Well, even before COVID and supply chain disruptions that allow people to extract, superior rents, we're in a very rarefied and specialized field. And I don't think the normal market mechanisms have fully taken hold to say, like, now some of these starting materials are more commoditized because there are more people producing it. Competition has done its thing and reduced those costs. So, before we get to the where we all want to get to, which is those bigger bioreactors producing things, there's work that can happen on the research side, as Shankar and I were just referring to, and then there's normal market mechanisms that may reduce the cost of some inputs. There's automation that will, I think that will support. I know that was one of the things that you mentioned in the prep document. All of those things will kick in. So again, it won't be any one thing, right? As this field matures, all of these things will kick in. We'll get better on the science. We'll get better. The supply chains will mature. Competition will take hold. Costs will come down. We should get smarter about drug development, which means we can hopefully take higher percentage shots on goal in precision medicine, so that we're not spending as much on clinical development. And ultimately, if we do all those things, then at the final point when we got to now produce something for an approved product, you will have accumulated less of the bloat that might happen because of failed product batches, high expensive inputs, poorly performing products, and unnecessarily large studies. If you can solve all of that, then eventually you've taken a lot of pressure off the manufacturing system to deliver magic at the finish line. We have to take a systems-based approach here to look at it and not think that manufacturing is critical. We've voted with our feet. We look forward to being being in the position that Josh and the folks at AskBio are one day. But there's a lot of other things that can and must happen to bring down costs in the system.
On the topic of supply chain, then, one of the things that we've heard about from various companies participating in this event is internalizing different elements of manufacturing at different stages, where either vector manufacturing, plasmid, other things. Are there elements of the supply chain that can be internalized in some way to either bring down the cost or the risks or some elements, or any other potential solutions to that element of the problem that you just outlined?
Josh, you're closer to the finish line. You want to take that first?
Yeah, go ahead.
You brought that up.
Yeah, so one of the things is, I think many people are starting to internalize their, making the raw materials. So from a transfection perspective, I know there's a lot of companies who are manufacturing their own plasmids instead of paying some of the higher costs at CROs. We did something similar by, switching to doggy bone DNA, which, helps us in terms of cost, as well as speed, right? So we can manufacture these pretty quickly in good amounts and then transition into tox studies or tox manufacturing, clinical manufacturing. And then, there's, in terms of supply chain, there's a lot of raw materials and supplies that have long lead time, and you need to understand what those expiration dates are and try to, stock your inventory, as well as maybe doing some of your own expiration studies to see if you can extend those shelf lives. It's a major concern, I think, for everybody. Single-use bioreactor bags, plastics. There's certain, tubing, things like that are impacting not only manufacturing but process development. If you're trying to re-engineer a new bag, you can't get the bag design. Get the bag designs, you can't get the materials, and so it's really slowing down really all efforts. And so it's kind of a lesson learned that everybody needs to learn from and modify how you look at your supply chain in the future. But right now you can try to do what you need to do, but you're probably not get ahead of it until you get some normalcy.
Yeah, we had a similar, I think it was sometime last year in the middle of the COVID. Supply chain issues existed already in gene therapy manufacturing because frankly, the supply for these drug products, for the supply of the clinical studies and the new companies formation, that was outstripping, the demand was outstripping supply. That was already happening. COVID only made things worse, right? So, we found that things that we could normally rely on, a turnaround time of a couple of weeks to months, then we were placing orders a full year in advance of our need, because that's what we needed to do to guarantee that we would have the relevant supply. We haven't chosen to internalize anything yet. We're not backward integrating to, acquiring any of the raw materials. I think the other thing, it's always, you never want to be a single sourced on any component. The range of things on which we now think about being dual sourced has probably grown because of the supply chain disruption. So place orders early, make sure that you're dual sourced up and down your supply chain. And also, hedge your bets. Josh mentioned bioreactor bags, and you might have gotten used to the idea that we're going to use vendor A's bioreactor bags, and we love their custom bags, and we're going to use those. And then you may find that vendor A may not be able to produce those exact bags or make them available to you because some COVID client is making, COVID vaccines is taking priority. So then you start exploring, it's like, oh, maybe we need to have bioreactor bags from multiple companies, and we need to understand the performance of our product and the productivity and the product attributes, irrespective of whether it's a standard bag or a custom bag or 200 liter, 1,000 liter, to provide maximum flexibility depending on what's available. So there are a lot of things that we went through, I'd say, last year to adjust to the new reality. None of it involves backward integration and internalizing it, but creating more flexible, more forward-looking and anticipation of our needs, and more dual sourcing and more flexibility to be able to pivot depending on where the supply chains are, so that you're not interrupting your production process.
I would just add to that. I think a lot of it relates to understanding your process. So, not flailing and really understanding exactly what you need, when you need it, at what scale and how much. And, I think the approach that we've taken here, and I think it's served us well, is really building from a platform process perspective. So, we have a pipeline of programs across multiple different therapeutic areas, but all of those feed into a common platform process that we built here at the company that is scalable and that is, roughly speaking, consistent across different portfolio products. And I think that is distinct from managing a portfolio that might include completely different types of products that have completely idiosyncratic manufacturing processes that require, customization, have unique bottlenecks, don't really give opportunities for scale. So I think that's something that the field probably needs to evolve in that type of direction ultimately, where there are common platforms that are scalable as opposed to unique one-off types of processes that serve only one product at a time, that allow for more predictable amassing of the necessary raw materials or other technologies that might be required for products.
So coming back to the cost and the implementation of all these different things that we've just talked about, under a blue sky scenario, let's say all of these innovations are implemented or cost saving measures are implemented, what kind of margins might be feasible in that setting? And Shankar, we can start with you.
Yeah, that's a good question. I mean, I think that at this point, as a field, we all have probably aspirations to move it in the same direction. And I think without sort of thinking about a specific number in mind, I think we feel it's possible to get margins in gene therapy along the lines of what we've seen with other biologics. So there is a possible future in which gene therapy margins could look like close to where antibodies were, or some iteration of how they've evolved over the last 20 years. That we'd hope that gene therapy margins could ultimately get to. And the ultimate goal is to think about how we can make this transformative modality that all of us have bought into as something that could change a number of different fields of medicine accessible to broader patient populations, where it's currently inaccessible to those populations, given very low throughput, extremely high cost methods of production. So it's not a specific answer to your question, but I think qualitatively, we think it can be directionally similar to where monoclonal antibodies have gone and have reached at this point today. Obviously, still a long way to go from getting there.
I'll take a different crack at the answer. I mean, margin involves two things. It involves price and cost. So I think, right, we have line of sight to how cost, and we spent some time talking about how costs may come down over time. Predicting price, I mean, many of us are here working with rare and prevalent indications. That's a fool's errand, and it's going to really depend on the therapeutic area you are. It's going to really depend on the product attributes that have to be really good. For a larger indication, you maybe be compared against a small molecule that is the standard of care. So we have to think about what the end goal is. What does your product need to look like when it is being used in the context of the patient population and is winning a prescription battle against something else that may not be a gene therapy? So you have to think about, so I would say if we started with the lens of we can get to margins that are like biologics, that could be good. But if you're comparing yourself to a small molecule and that's the standard of care that's doing a reasonably good job, then that might not be enough. So we really have to think hard about what is the end product. Like, I think it may sound crazy, but I think companies who are thinking of going into large indications, you really have to start with your target product profile. What does the end product need to look like at the finish line of a long development process, including, some hypothetical price, some hypothetical cost of goods, and can you get to your margins? And then you work backwards from there. And that might mean that you slow down your early work to try to take those extra steps, including on manufacturing and whatnot, so that you're ready for that final world, or what the world will look like for that product, maybe a decade from now. And that picture looks very different for larger indications, chronic diseases, versus for rare genetic disorder where the unmet need is very high and where there's literally no option. So you do have to think with the end in mind and then work backwards from there. And then what margins are, it's not a matter of what margins are feasible, it's sort of what margins are necessary in order for the product to actually work. And if you don't have line of sight into it, into how you're going to get to that price and get to that margin to make the business work and sustainable, then you may not want to start down that path, because this is not a build it and they will come, especially when you're dealing with large indications. We can't have that kind of thinking that we'll build it, it's cool, it's novel, people will pay for it. History, including recent history, is littered with examples of people who couldn't bring down their costs fast enough. Dendreon is a famous example of a cell therapy company, right, that was in the headlines for a while because it just couldn't bring down their costs enough to satisfy what the market needed them to look like, at least at the time of launch.
And I know we're kind of running low on time, and I don't think I can answer the question any better than Faraz and Shankar did. So I don't think I can really add more to that, more to what they already said. So I don't know if there's any other questions.
Great. So I think in that case, I think we'll turn to a couple of other questions regarding some of the considerations for larger indications. And so maybe from your perspective, when should a company decide to find a manufacturing partner versus developing capabilities internally? Obviously, each of you have talked about investments in integral manufacturing, but not every company could necessarily expand in a global footprint. So what are those considerations, especially for larger indications? And we can start with you, Josh, and then go to Shankar and then Faraz.
Yeah, I think from our perspective, we have a number of programs, and we have a platform manufacturing technology. So I think it was to our advantage, similar to Kriya and Tenaya, to internalize it. I mean, obviously, I know there's financing involved in all of that. I would say if you're going to outsource it and partner it, I think if you're a starting company, I think it makes sense. If you have a manufacturing technology, you don't have the infrastructure to tech transfer that to a CDMO and work with them to get into the clinic and hopefully raise some money so you can build it internally, depending on what your portfolio looks like. But I think for our instance, based on what we were trying to build, already having a platform manufacturing in-house, and AskBio has been around for over 20 years, and so we have cash coming in that was not through investments but through licensing and other collaborations and partnerships. So we were kind of in a unique situation where we could build out Virogen and get that up and running. So I think every situation is different. There's no right or wrong way to do it. I think the end goal, for no matter where you start, is probably to do that internally, so you have not only lower costs, but having that infrastructure, you can control your timelines. You're not always dealing with a CRO who's manufacturing a material, their supply chain, their analytics, and troubleshooting their analytics. You control most of that in-house, which helps move from bench to the clinic much faster. So I think the ultimate goal is probably where most of us are now currently. But I think you need to start somewhere, and you may need to partner with somebody early on. Now, post-commercialization, depending on what your footprint is, it may be good to internalize that or outsource that to a company that has the resources and experience for commercialization.
Yeah, I guess I can go next. I agree with everything Josh said, and I do think that internal, on balance, has huge advantages beyond what Josh described in terms of controlling supply chain and controlling your destiny. The cycle time to solve problems is infinitely faster, and also the learnings from those problem-solving exercise are to the benefit of the company solving those problems, and in the case of an internal problem solver, rather than having a CDMO learn those things on your behalf and use it to their benefit. So I think that there's a lot of advantages to internalizing. We have a big commitment at this company to data, and we've made significant investments in our computational platform and machine learning platform underneath many of those data sets. So I think there are unique things that you can do with data if those are internalized, within the company. But to Josh's point, it's also not feasible or practical for every company to go out and start right away and build out their own infrastructure and the team that's required to support that. So, it may make sense, depending on an area of particular focus, to partner with someone who knows, has the experience, has the infrastructure to do that. And it depends on what is the ultimate ambition of the company, whether it's to be an innovator in manufacturing or to be really focused only on the product and have a good partnership with someone who can provide expertise on the other side.
Yeah, in brief, I'll agree with what they just said, and I think it's in some ways it's unfair because you have three companies that all seem to have a very similar, we're all about big vision, maybe also going after rare and prevalent indications, novel capsid, novel platforms, we represent that end of the spectrum. But I think for those who are in the audience who are, I think your CMC manufacturing strategy has to follow your corporate strategy. So if your corporate strategy is actually not to try to take something all the way across the finish line and commercialize it yourself, but instead find a partner early on in development, your corporate strategy is to focus deep on research insight, but trust, and if the field has changed and the CDMOs, the capacity has come online, they're more reliable, they're able to produce, provide slots, with more flexibility and at lower cost. A lot of things could be, a lot of conditions could be different, and a lot of variations may exist in your corporate strategy that may make it completely the wrong thing to do, to internalize your manufacturing. And so I don't want, somebody who may be getting the wrong impression by hearing the three of us say that's the only way to go, right? Because it's not. You certainly can be more virtual in this. Even if you choose to be virtual, you just have to be thoughtful, right? You just have to be really clear on how important manufacturing is. And even if you don't intend to build it, you need to be very diligent, I would argue, preclinically about manufacturing strategy, partnership, and how that looks, even if you don't choose to internalize it. Because it's that important. You have to think of manufacturing as a critical aspect of your eventual product attribute, and then go out and find people who you can partner with and trust the future product attributes to them, right? And because you have to look at them as an integral part of your future product attributes, but you don't have to internalize everything. Certainly not.
Great. So with that, I think we're just about out of time. So I'd like to thank all of you again for the participation and for the great discussion that we just had.
Yeah, thanks, everybody.
Thank you. Enjoyed it.
Yeah.